Namaskara Karnataka, I'm Miss Gopika, your own biology master teacher. So a lot of students have been requesting me to teach outbreeding devices and about seed and fruit. So today's class is going to be a quick revision of outbreeding devices, seed and fruit. So let's directly get into the class. So all of you know that sexual reproduction in flowering plants, I have been doing it as a series of where I have done pre-fertilization event, post-fertilization events, and right now we are going to learn about seed and fruit, which is actually an overview of the entire chapter. I've not gone into line by line of NCERT because that is going to take a lot of time, and since you are students who are expecting or wanting only the high weightage chapters, this is the best way for you. So shall we start with outbreeding devices? So students, before I go to outbreeding devices, you should understand what is this terminology. So let me tell you, when a plant undergoes continuous self-pollination. Now what exactly happens during self-pollination? The stigma, the style, the ovary is the female part, and then we have the anther, right? So the pollen grains from the anther of the same flower is falling onto the stigma of the same flower. So continuous self-pollination leads to something called as inbreeding depression. So plants also have depression. Can you believe that? So plants also have depression, then how can we complain about our depressions, right? So students, when a plant is undergoing continuous self-pollination, it will go through a depression which is called as your inbreeding depression. But plants are very self-dependent people. They do not want to go to a doctor, or they do not have a doctor to go and tell that I am going through depression, can you do something? So to avoid self-pollination, so depression is happening because of continuous self-pollination. So to avoid self-pollination, plants are like, what? We only have steps to do this. Why do we even need to think? So that is when that outbreeding devices we are learning about. So to avoid inbreeding depression, right? To avoid inbreeding depression, they found outbreeding devices which will force the plants to do cross-pollination or also do show variation. Understood, all of you? So for those students who did not know what is outbreeding device, this is a quick recap, right? Continuous self-pollination leads to inbreeding depression. To avoid inbreeding depression, the plant will do some techniques, or it will take some steps which is called as outbreeding devices. So let's see which are the steps that the plant actually takes in to do this, okay? So students, the first thing the plant will do is change the position, okay? Change the position of the stigma style ovary, that is the carpel, and anther. That is, let's say if the pollen grains, okay, normally what is going to happen, let's draw a diagram to understand this. Okay, this is your style, stigma, and this is your ovary, and from here, okay, let's say our anther, right? This is the normal situation, a bilobed structure. So normally what will happen, when the anther matures, the pollen grains are going to land on the stigma, no doubt in that. So what did the plant do? Plant was like, I'm fed up of this, so let's do something, that is, I will change the size or the position. That is, the plant has decided to reduce the size of the anther or increase the size of the stigma, style, and ovary. Now students, you can imagine that the pollen has to go against the gravity, which is not going to be possible. So by changing the position of the anther and the carpel, the plant is going to force for cross-pollination to happen or gate-no-gamete to happen, but avoid autogamy as much as possible, okay? So this is the first ever step. The second step is going to be change the maturing. That is, if both of them mature at the same time, then the problem is going to be there. So if we change the maturation time, then it is also going to benefit in cross pollination, right? If the stigma and the anther matures at the same time, then only fertilization will happen, right? Then only pollination will happen. So changing the maturation time, that is, one anther is going to mature earlier or stigma is going to mature a little later. So maturation time is changed. Got it? Then the plant is like, let's do one more thing. See, stigma is the landing, okay, landing where the pollen grains will land. So what is going to happen when the stigma is going to get some pollen grains? This is the stigma landing place, this is the style, and this is the ovary is going to continue. Now let's say one pollen grain has fallen. The stigma is going to understand that this is compactable. So what is the stigma going to do? It is going to allow the formation of the pollen tube. So the plant has decided, let's do something called as self-incompatibility. Self-incompatibility. That is, the stigma will be like, you're from the same flower, you're rejected. You're from the same flower, rejected. So it is only going to take, okay, it is only going to facilitate other pollen grains, but cancel out the self pollen. Cancel out the self pollen. Understood? Self-incompatibility. That is making sure that these people are not going to be compatible because they are from the same place. Okay, they are from the same place, so we don't want them. Okay, these are few steps or few outbreeding devices. Then the last thing, okay, so students, basically 4 steps you have to remember. The last thing is formation of unisexual flowers. Formation of unisexual flowers. The plant will decide to form unisexual flowers so to avoid self-pollination from happening. In many cases, geitonogamy happens. Now, what is geitonogamy for those students who do not know? This is a plant. Okay, this is a plant. Now, on this plant, there are some beautiful flowers. Okay, some beautiful flowers are there. Okay, I'm drawing. Yes. So, students, some flowers are there, as you can see. Now, let's draw. Yeah. Now, let's say this is the male flower. Okay, this is the female flower. Now, what is going to happen? And again, this is the male flower. Now, what is going to happen? The pollen grains from this flower will automatically fall on this flower, right? So, that means on the same plant, it is happening between different flowers. Same plant, but different flowers. Okay. But same plant. So, geitonogamy can be possible, but again, geitonogamy also does not completely avoid the inbreeding depression, because again, it is just the same plant, it is just different flowers. So, again, variation. When we talk about these people's variation, it will not be equal to your xenogamy, or it will not be that great as a cross pollination. Understood, no? Just that you are marrying your atte ponnu, what mama ponnu, we tell, no, mama payya, atte ponnu, or like that means what? You're still in the same family. Your genetic disorders in the family will again come back to you. It is not like it is completely avoided, right? So here also what is exactly happening, students? Just because you're not marrying, like let's say a brother, sister, but still you're marrying someone from your family, that is, you're marrying your aunt's kid or your uncle's kid, still the genetic problems that are lying in the blood is going to come to all of you, right? So here also much variation won't be there, but it is better than autogamy, right? Better than, siblings. It's better than siblings, that this, it's going to another family, but still it will not maximize variation. Okay, shall we now learn about seed? So all of you know that the final product of your entire sexual reproduction has to be that the plant is not just used to expose or to show your emotions, even if you're sad, happy, you're crying, whatever it is. Flowers are not just for our greed. Flowers, or this angiosperms doing sexual reproduction, is ultimately where the ovary will form the fruit and the ovule will form the seed. So this is the ultimate destination for sexual reproduction to happen. So students, some notes I have put, so we will go through this. Now what is seed? So in angiosperm, the seed is the final product of sexual reproduction. And for those students who are thinking that, ma'am, I'm new to the channel, I just subscribed to the channel, I do not know anything about this channel, let me tell you, this channel is dedicated for all the KCET and board students from Karnataka who are appearing for KCET exam and who are appearing for 12th board, that is your PUC board examination. So you can go and check our previous lectures where we have done dedicated lectures for boards, which is under the video session, and then there are live classes as well. Every day live happens, students, from 6 o'clock till 9 o'clock, 1 hour lectures happen. It could be physics, chemistry, bio, it could be physics, mathematics. All your PCMB is right now in this channel. All you have to do is just press on that subscribe button and also share this, that this channel exists and this kind of teachers exist. And we are teachers who have been training NEET aspirants and JEE aspirants in our past, and right now also we are doing it through online mode. Before we have taught in offline. So we are very experienced teachers who have been preparing students for competitive exams. So I don't think you should be thinking that, are this teacher is going to help me crack KCET? Definitely, students, because KCET is easier compared to JEE and NEET. So it is easy for us to mentor you people and teach you better. Okay, so please consider subscribing to the channel, and this is for all our Kannada makkal out there. Okay, all the Kannadigas. I am born and brought up here, so I know the pulse at which you people are studying and what is happening. So bega video na like ಮಾಡಿ, subscribe ಮಾಡಿ channel na, ಆಮೇಲೆ share ಮಾಡಿ friends ಜೊತೆ, because it's going to help us a lot. Okay, coming back to the chapter. So students, the notes will be available on the Telegram channel, which is We Karnataka, same. Okay, the channel name only you can just type on Telegram, and the link is in the description box, so you do not have to search anywhere. Okay, We Karnataka, KCET and boards. And if you want to join my Telegram channel, that is biology by Gopika. Okay, you know my name, Gopika. I'm your biology master teacher. Okay, so these are the two Telegram channels where all the notes will be available. Okay, yes. So students, in angiosperm, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Okay, so as I told you, ovule has to end up forming the seed. Now seeds are formed inside the fruit. All of you know that. Okay, a seed typically consists of a seed coat. There will be a seed coat, and the seed coat is actually formation. The seed coat has been formed by your integuments. How many of you remember integuments? Remember when I taught you embryo, I taught you integuments, inner and outer integuments, which are going to do the job of protection. So those people have modified to become your testa and tegmen, which is your seed coat. Okay, cotyledons, right? When you open the seed, you see cotyledons, right? This is the cotyledon and an embryo axis. So as you can see here, students, there is an epicotyle, okay, hypocotyle, a radicle, which is going to end up forming the root. Okay, root. And then your cotyledons, which is this surface, and your seed coat, okay, and your seed coat, which is made up of your testa and tegmen. So this is what your seed structure is. The embryo is made up of a radicle and an embryonical axis or two cotyledons. So it can be monocot or dicot, which you will be studying later in your botany chapters thoroughly. When you're studying, when you have studied your 11th morphology, you would have learnt all this, your cotyledons, monocots, dicot seed. But here you just have to know overview of it. Okay, so the embryo is made up of a radicle, an embryonal axis, and one cotyledon as in wheat and maize, two cotyledon as in gram and pea. All of you have seen, no, green gram. Okay, then we tell, no, moon dal, tur dal, all these dals also you would have seen. Now if you open your green gram and see, you will see two cotyledons, right? So here, like this, two cotyledons you will be able to see. So students, as you can see, the same thing, only testa, seed coat. You can see embryonic axis. Okay, embryonic axis is here. And you can see the radicle, which is going to form the root, and your plumule, which is going to form the shoot. Okay, all this is just a general understanding. So embryo has cotyledon and embryonical axis. Embryonical axis will have your plumule, epicotyle, your point of attachment of the cotyledon, hypocotyle, and radicle. Okay, we'll come into that, don't worry. Now, the cotyledons of the embryo are simple structures, generally thick, and swollen due to storage of food. So everywhere the plant makes sure that the food is stored, right? When we spoke about embryo, we heard about endosperm, who is going to act like the nourishment is going to be provided to the embryo. When we spoke in the when the growing embryo stage, we spoke about nucellus, who is going to give the nourishment. So when you see that, no, the plant always makes sure that the nourishment is provided so that they do not have to depend anywhere. So here also same thing. The cotyledon region is where the storage of food is going to happen. So as you can see, this portion is going to store the food. Okay, this portion is going to store the food. It could be a dicot or it could be a monocot. Monocot means one cotyledon, dicot means two cotyledons. Okay, so thick and swollen due to the storage of food reserve, as in legumes. Mature seeds may be non-albuminous or albuminous. Now, as I already mentioned to all of you, endosperm is going to provide the nourishment for the growing seed or the embryo we tell, and then later the seed. Now, in some seeds, the endosperm will be fully consumed. In some seeds, the endosperm will be left a little. So, depending on that, we have two types of seed. You can call it albuminous, non-albuminous, or endospermic and non-endospermic, right? So, here you see albuminous seeds, okay, very, very important. Endosperm is not completely used, okay, used by the developing embryo. So, a portion of it remains in the seed. Okay, example is going to be, students, coconut, castor and maize. Okay, so remember like this, albuminous seed is the CM. Okay, is the CM. CM. C for castor, M for maize. Now, your MCQ questions in your KCET can come depending on the example names. So, be smart enough to learn the example. So, remember, albuminous seeds are where the endosperm is not completely consumed. Some of it is going to be present in the seed, and the example is going to be CM, Chief Minister, castor, coconut and maize. Okay, now non-albuminous endosperm is completely used up by the developing embryo, and the seed will be left with no endosperm. Okay, no endosperm left in the seed. Example is going to be pea, bean and mustard. So here you are going to remember PM. So students, PM and CM is having a competition. Okay, P for pea, okay, M for mustard. So, CM, PM fight is going to happen, right? That is albuminous and non-albuminous. He is telling that I have some of the money that was given to me that I have not utilized, so that I am going to take it to my family. PM is telling, no, I do not have any money, that I do not have anything to take it back to my home. So, CM and PM is going to fight for food reserve or the nourishment that they were given with. So, endosperm is left albuminous, endosperm is not left, non-albuminous. Non means what? Nothing is there. Okay, so is this clear? CM and PM. Okay, that war is going to be CM versus PM. Okay, here endosperm, that means CM has some money, PM has no money. Okay, so very suspicious conversation we are having. So seed, occasionally in some seeds such as black pepper, this is a very important question, and black pepper and beet, okay, remnants of nucellus are also persistent. So students, here basically we are talking about all the food reserve. Like I already mentioned to all of you, nucellus was the one who was giving the nourishment for the growing embryo sac. Right. So all of you remember the ovule structure, no? This was how your ovule was there, integuments was there. After the integuments were present, you remember inside this embryo sac was there, here nucellus was there. They are telling that in some of them, okay, remember B square or BB, okay, remember that in black pepper and in beet, okay, some remnants of your nucellus is there, and we call them perisperm. Okay, so this residual persistent nucellus is the perisperm. So when nucellus is going to be staying for a longer time, nucellus would have got over, or all of that we would have consumed when the embryo was growing. Okay, after that we gave embryo the endosperm also, but this nucellus did not go. Nucellus is like, I am very persistent. Yeah, you people also should be persistent during your KCET preparation, no matter what comes. Okay, ಏನಾದರೂ, okay, whatever comes, make sure that you're persistent, because if you're persistent and if you have perseverance, life is going to take you somewhere else. Okay, so your dream engineering college might be, students understand, dream engineering college just does not mean that, oh my god, my name is going to be glorified, I'm in RV engineering college, I'm in this college. It's not just about the name. It's that your friends are going to be that type, your ambience is going to be that type, your lifestyle will upgrade. If you be with people who are like always thinking about what to eat today, or how do I survive the day today, I do not want to live, you're always going to be like that. But imagine people who talk about business, who talk about startup, who wants to upgrade their life, who thinks about, exercising every day, who thinks about, meditation is important, reading books. Your lifestyle is going to be so different. So dream medical college, persistence is going to take you. Okay, so persistent nucellus is going to be called the perisperm, and perisperm is B square, that is beet and black pepper is the example. So examples are very, very important. Drink some water during the entire class. Perfect. Moving on. Now, perisperm and endosperm. It is present where the persistent, okay, nucellus of the ovule is there. Understood? It develops from the primary endosperm nucleus. So people who don't know primary endosperm nucleus, please run to my previous class and see when I spoke about double fertilization. I taught you PEN thoroughly. Okay, it is a part that belongs to the seed. It contains reserve food material. It is usually dry. It is usually in the fluid form or soft material. Example is black pepper. Here example is going to be water of coconut, pea and beans. Okay, pea and beans. So students, the water that we drink, from the coconut, tender coconut, Elniru we tell, no? Elniru's, that water is actually endosperm. Okay, it's going to be the endosperm. Clear? Yes. Now seed. Integuments of the ovules harden as tough protective seed coat. The micropyle remains as a small pore in the seed coat. So those students who remember the entire ovule structure thoroughly and remember the embryo sac will be able to remember that there was a small micropylar portion that actually is right now also there in the seed, so that some exchange can happen. Same way your integuments have modified to form seed coat. Okay, clear to all of you? Micropyle facilitates the entry of oxygen and water into the seed. This is a very important question. Okay, mark it right now in your NCERT and keep. Micropyle region was there, in the ovule, that will actually continue in the seed, and that small portion is going to allow exchange of oxygen and water. Okay, you can see here, this is going to help in some oxygen and water exchange. Oxygen and water exchange. As the seed matures, its water content is reduced and the seed becomes relatively dry. Okay, the general metabolic activity of the embryo slows down. The embryo may enter into a state of inactivity, okay, called as dormancy, or if the conditions are favorable. That means, students, let's say the seed has fallen on a rock. Okay, now in the rock, the condition is not favorable. The seed will go into a complete dormancy, let us sleep. Okay, seed will be like, I'll chill, yaar. I don't want to germinate because my conditions are not favorable. But the moment conditions get favorable, the seed will start germinating. Okay, that is what they're telling here. Now, as the ovules mature into the seeds, the ovary develops into the fruit. And the transformation of ovules into seed and ovary into fruit proceeds simultaneously. It's not like, okay, let the ovary become a fruit, then the seed, ovule will become seed. No, both will happen together. When you're eating a fruit, fruit is also there, seed is also there. If you're eating a fruit, when you're halfway, the seed will be like, wait, I have to be growing. It is not like that. No, both will happen simultaneously. Okay, the wall of the ovary develops into the wall of the fruit. Very, very important. The wall of the ovary develops into wall of the fruit, which is called the pericarp. So as of now, you have learned so many things that you have to remember. Remember that your integuments, okay, integuments have become seed coat. Okay, seed coat. Persistent nucellus. Okay, persistent nucellus has become perisperm. Okay, persistent nucellus have become perisperm, and let's write the seed fruit coat. Okay, so ovary wall has become pericarp. that is your fruit wall. Okay, fruit wall. So these changes have happened, and then your micropyle. Okay, micropyle that was there, it is going to help in exchange of O2 and H2O. Okay, yes, this is what you have learned so far. Okay, so here simultaneously is happening. Generally the fruit consists of a wall or a pericarp and seed. The pericarp may be dry, may be fleshy in some. When the pericarp is thick and fleshy, it is differentiated into an outer pericarp, okay, epicarp, and the middle mesocarp and the inner endocarp, which you have learned in your morphology of flowering plants in class 11th. Okay, that is, your pericarp can be divided into 3 categories, that is your epicarp, mesocarp and your endocarp. Okay, inside endo, outside epi, middle meso. Same thing we study, mesoderm, endoderm. Okay, same thing. So here students, in most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off because it's not required, right? Now, let's say your exam is over. Will you still keep the book with you, telling that, oh my God, I really have to keep this book, yeah? Sometimes you will be like, no, let's give it the book, right? So, after the fruit has developed, right, most of the other parts will degenerate. However, in a few species such as apple, very, very important, few species such as apple, strawberry, cashew, etc., the thalamus also contributes to the fruit formation. Such fruits are called as false fruits. So, students, normally ovary should become the fruit. Do you agree to all of this? Yes, ma'am, ovary should become the fruit. But they are telling that other than the ovary, the thalamus is becoming the fruit. If I am telling you that you are a KCET aspirant or you are wanting to crack KCET because you have registered for it or you know everything about it, you are sitting somewhere and you are thinking that yes, I am a KCET aspirant, but you don't know anything about KCET, you don't know when registration is going to happen, you don't know when the exam is going to happen. Can I call you a true KCET aspirant? No, I will call you false, fake. Okay, fake I will call you. Same thing here. If ovary is going to form the fruit, these kind of fruits, okay, ovary is going to form a fruit, I will call it true fruit. But if thalamus is going to form the fruit, I will call it false fruit. Because what has to happen that did not happen, something else happened. So I cannot tell that that yes you are still a fruit. No, it will not happen like that. Clear to all of you? Yes. So students, false fruit is formed when other than the ovary, any other floral parts, okay, help in the formation of the fruit. We will call it false fruit. So the apple that you're eating, next time you go tell mama that you're eating the thalamus, she'll give you one on your tale. Okay, she'll be like, what? I'm eating an apple. Okay, so, but the portion that we are eating is actually the thalamus that has modified to become a fruit. Okay, yes. Most fruit, however, develop only from the ovary and are called as true fruits. Clear? So, fruits are divided into 2 main categories, true fruits and false fruit. True fruits is the one that develops from the ovary. False fruit, any other floral part other than the ovary will form the false fruit. Okay, yes. So that is all about your fruit as a whole, but I would like to add on to one more thing here. Since we are in this topic, I would like to add on to some more things here to all of you. So students, so as I told you, fruits, when we categorize, okay, fruits when we categorize, first we will call true fruits like your mango, okay, and we will call false fruits like your strawberry and apple. Then there is something called as parthenocarpic fruit. What is it? Parthenocarpic fruit. What is parthenocarpic fruit? The fruits which are formed by not undergoing fertilization. Now I told you that everything should happen properly, right? Pre-fertilization, fertilization, post-fertilization events. But here, parthenocarpic fruits are the ones where fertilization will not happen. Fertilization does not take place. Fertilization does not take place, but the fruit is going to be formed. Clear to all of you? Students, same way, you have one more topic in your NCERT, that is apomixis. That is where seeds are going to be formed. Seed formation. Okay, let's write the whole thing. Formation of seed without fertilization. Clear to all of you? So, fruit is formed without fertilization is called parthenocarpy, and seed without fertilization is called apomixis. Okay. Then one more topic in your NCERT that would cover your entire chapter is your polyembryony. Polyembryony. That means, students, we all know one embryo is going to be there, but what happens? Some of the nucleus cells will come inside, they will move to inside and they will form multiple embryos. So when like that it happens, that is multiple embryos, multiple or many embryos are formed, we call this condition as polyembryony, which is seen in your citrus fruits. Okay, so example is going to be citrus fruits. Yes, students, examples of all of this try to remember as much as possible. Be it here also you can write mango. Okay, here you can write apple or strawberry. Okay, here you can write banana. So these examples, if you can remember, it is well and good, because these are going to be the topics that are going to be asked as questions, because these are all little exceptional topics. No, now we know true fruits are the ones who are, I will also write that. Okay, true fruits, where it is, what is going to happen? Ovary will form fruit. Okay, ovary will form fruit. In false fruit, what is happening? Thalamus or any other floral part. Okay, thalamus or any other floral parts are helping in the formation of fruits. Here what is happening? Fruit is being formed. Fruit formation without fertilization. Okay, without fertilization. And here what is happening? Seed formation is happening without fertilization. Clear to all of you? Polyembryony, what is happening? Many embryos are going to be there, and that is going to lead to multiple embryos or many embryos, like in citrus fruits. Okay, so here this is happening because the nucleus would have come inside, moved inside. Okay, when the embryo formation is happening, no, we all know inside the ovule the embryo is going to be there, right? So when that formation happened, the nucleus would have moved inside, because of which the multiple embryos are formed, and when multiple embryos are there, we call that as polyembryony. Okay, I will write it a little down, otherwise it will not be clear to all of you. Okay, polyembryony. Polyembryony, many embryos. Okay, students, example is very important, so try to remember the examples. Okay, citrus fruits. And one more small topic that we did not cover in the previous things are your artificial hybridization. Okay, artificial hybridization also is a very easy topic. Okay, nothing much to remember. So students, artificial hybridization means where we want to make sure that, let's say, what we desire, right? What kind of flowers we want or what we desire in the plants, we will try to make that happen with two steps, which is called as bagging and emasculation. So first I will write emasculation. Okay, emasculation and bagging. So here what is happening, students? Emasculation means where we will cut out the anther. So what we are trying to avoid is, we do not want self-pollination to happen. We do not want pollen grains to fall on the stigma. Instead, we want to dust the pollen grains of our choice on this plant. Okay. So here what we are going to do is, we are going to remove, okay, remove the anther using forceps. Okay, we are going to remove the anther, and after removing the anther, we have to make sure that this plant, okay, let's take, this is the, okay, this is the plant and this is the flower. So from this flower, okay, this is the flower, this is the flower. In this flower, let's say our anther is there. Okay, we are going to, first step, we are going to remove this anthers. Okay, we are going to remove these anthers. Okay, we're going to remove these anthers. So once we remove these anthers, students, what is going to happen? We have to take this flower and make sure that this flower is going to be covered, okay, because anther got removed. So we are going to cover it with a butter paper or a polythene paper like this, and this is called as bagging. This is called as bagging. Okay, so bagging is the process of covering the flower, covering the emasculated flower. Chumma flower, if we cover, what is the use? Covering the emasculated flower. Now after covering this, we will make sure that there is still the stigma, right? So let's say our stigma is here. Okay, stigma is here. To this stigma, we will make sure that we dust the pollen grain of our choice. So after this is done, we will make sure to dust, okay, dust the pollen grains of our choice or desire onto the emasculated flower. Clear? So this way what happens, I am going to get the desired, what I want, I am going to get. Unnecessary pollen grains will not be dusted on this. So we remove the pollen, bag the flower. After that, we bring the pollen that we want, dust it on the stigma, let it compatibility. Again, we are going to bag it, because again, unwanted pollen grains should not fall on this. Understood, all of you? So the next process of this is going to be where the flower is going to be removed. Okay, bagging is going to be done. After that, the flower will be let, okay, the flower will be dusted with another set of pollen grains. Another set of pollen grains will fall on the stigma. Okay, another set of pollen grains from this will be dusted on the stigma. Clear to all of you? Yes. This is your emasculation and bagging, which is a part of your artificial hybridization. This is also done so that we get what we want, right? Favorably or desired flowers and plants will be formed, right? This is done by a lot of people, as you know, like how tomato is formed, tomato plus potato, like that. So when we want to make hybrid, or when we want to make our desired plant, this is called as artificial hybridization. Okay, so all the topics of your sexual reproduction is covered, students. Fruit, seed, pre-fertilization event, post-fertilization event. Please check out the video. I would like to open this, one second. Just give me 1 minute, and I think the video will pop up here. Okay. Okay, 1 second. Let's search. Yes, it's here. We Karnataka, KCET and boards. So students, this is the channel that you're watching. Okay, if you go to this, if you go to the live, okay, and you go right down, okay, you go right down, you will be able to see, all the lectures here. See, molecular basis is done. Okay, same way, if you go further, again, molecular basis, structure of DNA and experiments are done. If you go further down, you will see sexual reproduction in flowering plants, where I have done the whole chapter. So here you can see I started with, okay, your microsporogenesis, your anther, stamen, everything, and then later we came up to your double fertilization. Okay, I've also spoke about pollen pistil interaction, and then we also learned about your entire pole and pistil interaction plus double fertilization. Okay, so beautiful notes are also there. So all you have to do is subscribe to the channel, students, and as you can see this is the video. If let's say if you're watching this video right now, just go here, click on the link, and automatically the Telegram channel will open for you. Right now we have around 500 students in the Telegram channel. So I hope this video was helpful. See you all in next class. Till then, tata, bye bye, see you all, take care.